Greetings everyone, and welcome to NACFC 2020 and the first plenary. Well, this year and this meeting look different than any other in our history. It is great to be together, and I have no doubt that this year's meeting will once again inspire us and re-energize us in our fight against CF. 2020 has certainly not turned out the way any of us expected, but as I look back on the past year, I have such tremendous gratitude to all of you for the work that you have done to tirelessly advance CF science and care and serve people with CF and their families. Thank you. In keeping with this trademark relentless spirit, the CF community has already risen to the challenges of a virtual scientific conference during a global pandemic. It's been exciting to see over these past few weeks the number of people who have taken advantage of the early access to NACFC material and begun exploring all of the available content. We have thousands of people joining us online today for this plenary presentation. And like you, I'm looking forward to the live discussions and Q&As over the next few days. I also encourage you to take time to virtually visit the poster presentations and the supporter showcase. Now, last October, we gathered in Nashville to celebrate 30 years of progress since the discovery of the CFTR gene. It was actually 1 year ago today that Trikafta was approved by the FDA. This past year, we've had the opportunity to see the life-transforming effect of highly effective modulator therapy on the lives of so many with CF. We've also seen progress toward the expansion of highly effective modulator therapy to even younger age groups and to some of those with rare mutations. I think we can all say with confidence that we have entered a new era in CF. But at the same time, we also know how much more work we still have left to do. Even with highly effective modulator therapy, people with CF are still battling every day the challenges of their disease, and too many are still waiting for their breakthrough. That is why over this past year, we've put so much energy into our infection research initiative and into our growing programs for advanced lung disease and lung transplantation. It is why this year, for the first time in our history, we will have an NACFC plenary focused on the GI manifestations that continue to challenge many with CF. It is why at the CF Foundation that throughout these challenging times, we've been working in partnership with care centers to help support continued delivery of outstanding clinical care and working closer than ever before with people with CF and their families to find new and innovative ways to navigate the complexities of daily life with CF. Now, in a few minutes, you're going to hear from Dr. Brian Davis and Dr. Maria Limberis about taking on perhaps the most important area of this work that we still have left to do, assuring that all people with CF have a treatment for the underlying cause of their disease, no matter what their mutations. Now, we're just beginning to lay the foundation for this work, which we call Path to a Cure. It was at last year's meeting that we announced the $500 million Path to a Cure program to accelerate progress in this all-important area. We know this is a challenging task and is not a simple undertaking, but just as we overcame the notion that using small molecules to refold a mutant protein was too difficult, so will we overcome the notion that genetic therapy of CF is too difficult. The CF community has a long history of being successful and taking on challenges that seemed insurmountable. Let's let the challenges we are facing this year be just the next ones in line as we continue to move forward to assure all people with CF have the opportunity to live long, fulfilling lives. Now, I'd like to turn the program over to Dr. Bill Skatch, the Chief Scientific Officer at the Cystic Fibrosis Foundation.
Thank you, Mike. Welcome, everyone, to the first plenary session of the 2020 North American CF Conference. My name is Bill Skatch, and it is my privilege to introduce today's plenary speakers. Last year, we announced the Path to a Cure, with the goal of accelerating efforts to treat the basic defect in 100% of people with CF and ultimately develop a cure. This is our largest and in many ways the most challenging program that we have ever developed. To accomplish our goal, we will need to bring in new technologies for treating genetic diseases. And to do that, we need to lay the groundwork, create the science, and provide incentives and tools for companies to successfully develop the CF therapies of tomorrow. Today's plenary speakers are the perfect individuals to provide an update on the exciting progress that has been made and to lay out the roadmap for success. We are living in a very different world today than we imagined a year ago. And like everything else, CF research has had to adapt to the realities of COVID. I can only thank and congratulate the entire research community and our industry collaborators for their resilience and the amazing progress that has been made under very difficult circumstances. Today, you will hear about some of the groundbreaking discoveries that in just the last year have provided an unprecedented insight into the biology of the CF airway, information that is already helping scientists and developers work together to devise new ways to target cells for genetic-based therapies. The path to a cure has also stimulated a groundswell of interest from industry to apply their cutting-edge technologies to CF. We are currently working with nearly 40 companies in the path to a cure alone and have already funded 15 industry programs. We expect a clinical trial for nonsense therapy to begin this year, and the results of the first mRNA therapeutic trial in CF are expected in 2021. We have provided up to $14 million in additional funding to 4D Molecular Therapeutics to accelerate their gene therapy program. And in just the past few months, we created a $20 million CF incubator in collaboration with the Boston venture-based group Longwood Fund to build CF-focused companies from the ground up. This is an entirely new type of program for us, and we are very excited about the possibility that it will provide to the CF community. In our first presentation today, Dr. Brian Davis will tell you about new discoveries in stem cell research that are providing key information to inform genetic-based therapies. Dr. Davis is professor and director of the Center for Stem Cell and Regenerative Medicine at the University of Texas Health Science Center in Houston. However, his path to CF research was not a straight line. He received his bachelor's degree in physics, graduating summa cum laude from Harvard University, and then a PhD in physics from the California Institute of Technology. Over his career, Brian's research has spanned many areas, including viral causes of cancer, pathogenesis of HIV/AIDS, and identification and characterization of bone marrow stem cells, and he was one of the early pioneers in gene editing. Much to our benefit, he has also become a true scientific force in CF, where his research is focused on genetic correction of CF lung stem cells and therapeutic advances for people with cystic fibrosis. Our second speaker, Dr. Maria Limberis, will then provide an update on strategies for delivering genetic-based therapies to the lung. Dr. Limberis is a research associate professor in the Department of Medicine at the University of Pennsylvania, and she has worked on genetic therapies in the lung nearly her entire career. She received her bachelor's degree in with honors in biochemistry from Liverpool John Moores University in the United Kingdom, and her PhD degree in molecular biology from the University of Adelaide in Australia. There she studied lentiviral-based gene therapeutics, which then moved her to the University of Pennsylvania for postdoctoral studies in the laboratory of Jim Wilson, where she investigated novel airway-directed gene therapies for CF. She has established herself as an expert in viral-based gene delivery with development of an innovative vaccine strategy that is now being developed for influenza and more recently for COVID-19. But before we hear from Brian, I just want to highlight why the path to a cure is so important. We are incredibly excited about the recent progress in CF drug development, but we are acutely aware that more work is needed so that everyone in the CF community can look to the future with equal optimism. We never forget that not everyone can benefit from CFTR modulators, and we will never be satisfied until there is effective treatment and a cure for everyone with CF. I don't think there's a better way to portray the urgency of our mission than with the following video that provides just a glimpse into the hopes and dreams of an inspiring young woman named Julia.
I show my dog in 4-H, and last year we got overall champion. It's so amazing to win something like that because cystic fibrosis can take so much from you. It can be nice to just pretend that you're just like everyone else. And it's just little things like that. When you win, it's so much more gratifying than if you were to win any other way.
People tell us all the time that they can't believe that she's fighting this horrible disease because you would never know it by her demeanor. She's always laughing, telling jokes. She has no issues getting up at 4:45 in the morning for swim practice. She just is one of the strongest people I know that's overcome some pretty serious challenges for her age.
Say hi.
So Julia has what they consider class 1 or nonsense mutations. We have actually had our medicals clinic, CF clinic, swab her nose and take her nasal cells and test these new drugs that are coming out. But as we expected, we didn't see any improvements.
When I see other kids with CF getting the modulators and seeing them work and improve, it warms my heart. It makes me so happy to see them get that happy ending. At the same time, it does break my heart a little bit because I know that those modulators won't work for me and I won't be able to get those results. My biggest concern as I get older living with cystic fibrosis is that I'm a little bit afraid of the responsibility that comes with it. If I'm exhausted from a long day at school and I just don't feel like cleaning my nebulizers, my mom will just understand and she'll pick it up for me. But if I'm on my own, I won't have that lenience, and it's a little scary.
When I think about the future of Julia with CF, sometimes it becomes overwhelming. The what ifs tend to get to me. One of our friends saw Julia doing a breathing treatment and said, "How long does she have to do this for?" I said, "Well, the treatment lasts about 45 minutes. She goes, "No, I mean, how long?" And I said, "There isn't a cure now. She will do this forever." If there becomes a treatment available for Julia and her type of nonsense mutations, those what ifs go away. And learning about how to cope with life in that way is a challenge we highly welcome.
To everyone who does contribute to the research for CF, just thank you. I know for a fact that I wouldn't be here today without you. The dream for me would be to have a great job, having a family and a big house with a wraparound porch. Even if I do see myself like sitting on the front porch doing one of my best, it's not really a sad thing. It's just sort of a thing that I think about when I think about my future, because it is a part of my life now. But just imagining not having to do my treatments and having all these great things is just awesome. It's truly one of the best things to think about.
Wow, what a powerful story. It's easy to see why we go to work every day and why the entire CF community is working so hard to find a cure for this disease. And now, please welcome Dr. Brian Davis.
Bill, thank you very much for that introduction. What I'd like to do is welcome everybody today to this first plenary in which Dr. Maria Limberis and I are going to seek to lay a foundation for an initiative announced last year at the NACFC in Nashville for the path to a cure. And what we're going to be focusing on today is really what are the fundamentals of genetic-based therapies to be able to restore CFTR function, in particular to the airway. So as I just mentioned last year in the introduction to this major focused initiative, Path to a Cure, it was indicated that the thrust is to develop a cure for everyone with CF, irrespective of their CFTR mutation. And a schematic that was presented is shown here, where on the left we show essentially what we're dealing with are our mutant or mutated CFTR genes. CFTR is the protein that is defective in cystic fibrosis. And as mapped out here, moving from the top to the bottom, there was first the possibility of just repairing the protein. The second was restoration of protein expression. And the final was to either fix or replace the CFTR gene. And today we're largely going to be focusing on the latter two, that is the restoration of protein and the fixing or replacing of the CFTR gene. So shown here is a schematic really in the case of a normal CFTR gene. And we're going to work our way from the bottom of the slide to the top, where at the bottom we show in the chromosomal DNA, the CFTR gene, the DNA, and then that is then specifically converted into what we call messenger RNA. This is a single-stranded molecule that basically encodes for the full CFTR protein. That protein then moves into the cytoplasm where it's acted upon a normal cellular machinery to give rise and synthesize the CFTR protein. When that protein is normal and not mutant, it then naturally moves to the surface of the cell and then gives rise to functional CFTR channel activity, which allows for anions such as chloride or bicarbonate to move in and out of the cell and maintain hydration of the mucus layer, which you can see in green above. Now, for cells that are present in the airway, in fact, the surface of the cells is at the surface of the airway. So it really restores that functional capability to cells of the airway and other organs as well. So let's look at what takes place in the case of CFTR mutations. So let's start from the bottom of the slide and move up, where the bottom of the slide, we actually see the CFTR protein. And you can see identified regions, TM for transmembrane, MBD1 for nucleotide binding domain 1, for example. And these are highlighted in specific colors. If we then move up to the next level, the middle of the slide, this is where really the coding sequences are encoded by the messenger RNA. And you can see that very specific regions of those domains, for instance, the yellow or the red, are represented by certain regions of that messenger RNA. And again, that's what gives rise to the full normal CFTR protein. When you go one level up, it turns out that the way that this information is actually encoded in the chromosomal DNA is not compacted into that 4,400 bases, for example, but it's actually spread out over a much larger region, 250,000 base pairs. And importantly, it's the coding sequences. We call them exons in this case, 1 through 27. They're actually separated by specific sequences that get spliced out or removed in the process of generating the appropriate messenger RNA. One can have mutations that affect various processes in this. One in the case is stop codons, which can actually stop the appropriate synthesis of the full-length protein and give rise to a shortened protein, which is then normally degraded within the cell. So there's little to no protein that's present. Also, there's a possibility that there can be mutations that affect splicing, and what you may see is up on the upper panel, just around exons 11 through 12, you'll see a particular mutation that's 17:17-1 mutation. And that's a case where it actually affects that splicing function so that one doesn't remove those specific, what we call intronic sequences. And this gives rise to a messenger RNA that no longer properly encodes the appropriate protein. So I'm just going to summarize in this slide here that we have what we call these premature termination codons. Those are the ones that cause the stop. That is, as opposed to encoding a glycine amino acid, in fact, we have a stop and the translation, the synthesis of the protein stops there. That's a whole class of proteins that gives rise to no CFTR protein. So to really restore CFTR protein, we have to restore either the DNA or the mRNA to encode the full-length protein. In the case of the splicing mutations, they also give rise either to no protein or an aberrant protein. So that's a case also where through our genetic-based therapies, we need to restore the full correct sequences. Now, even though these classes of mutations are the ones that are initially going to be addressed through the genetic therapies, essentially we want to develop these methodologies to eventually be applied to all CFTR mutations. So we will start with those mutations that do not produce protein, but eventually they may be applied for long-term correction of all mutations. So we've talked a little bit about mutations. Let's now talk about at least one of the major organs and tissues that we really need to address for restoration of CFTR function. And in this case, we're going to talk about the airway. Shown on the left-hand panel is really a cross-sectional view of a region of the human airway. And what you can see is essentially a number of stained nuclei in that dark purple. And you can see at the very top what we call cilia. These are hair-like molecules that are involved in moving mucus. So to get a better idea of what's going on, this schematic on the right will try to identify at least 4 of the major cell types that are involved. The very far right, we see a secretory cell, and those essentially within the cell there are there's mucus, which then becomes released from the cell, coats the surface of the airway, and that's what's involved in actually moving contaminants and pathogens up and out of the airway. You can also see highlighted on the surface of that secretory cell is in green the evidence of CFTR channel. That's actually what's involved in the passage of chloride or bicarbonate and is required for normal functioning of a cell. If we move to the far left, you can see the ciliated cell, and this is the cell that has those sort of hair-like structures on the surface, and that's actually the motion of which moves the mucus up and out of the airway, and therefore all the contaminants or pathogens that are trapped also move in that way. You'll see at the very bottom of that schematic what we call the basal cells. And these are cells that play a very, very important role in the ongoing regeneration and repair of what we call the airway epithelium. That those cells that are directly exposed to the surface, and the surface airway would be the very top, right above where those cilia are, that those cells are being exposed and damaged either through normal aging or exposure to inflammation or other pathogens such as viruses. So the body has a natural way of actually restoring the structure and the architecture of those cells by the basal cells dividing, proliferating, and then differentiating or turning into the more specialized cells that you see above. So those cells play a very important role in the airway. And finally, I'm going to identify a cell which only was recently confirmed through one of the methodologies I'll be talking about next, the ionocyte, that although a very, very rare cell, this is a cell that expresses high levels of a messenger RNA for the CFTR protein and actually is believed to play an important role in normal function and then in the case of CF disease. So that's sort of an overview, and I would like to basically draw your attention now to recent methodologies that allow us to understand in far greater detail the composition of cells of the airway, their level of CFTR expression, and how those cells may differ between, let's say, a non-CF case or control and that case of CF. And for this purpose, what I'm showing here is just one report that was funded by the Cystic Fibrosis Foundation, the consortium, Epithelial Stem Cell Consortium, investigators from UCLA, Cedars-Sinai Medical Center, and the CFF laboratory up in Lexington, Massachusetts. And what you can see on the left-hand side is sort of this architecture of the airway. And the colors that you see there are, in fact, regions of the airway that were specifically dissected out by these groups of investigators. And then they applied to it a very, very powerful new technology that's called single-cell RNA sequencing, which the cells of the tissue are dissected into single cells, and then for each of those single cells is quantitatively analyzed for what messenger RNAs, what genes are being expressed, and at what level. And this allows us not only to identify cell types, but to see how those cells may differ between non-CF and CF. And just as a summary of these data, let me take you to this next slide here. And this is very, very complicated. I'm not going to really emphasize too many features of this, but let's just sort of go to the center panel if we can. And let's look at essentially the cells that are stained somewhat at different hues of green, for example, at the bottom. And what this just shows is these different hues of green, in this case, 5 different clusters, all identify basal cells, but different types of basal cells that can be differentiated or distinguished on the basis of what types of mRNAs are being expressed. So we now have a much clearer idea of sort of the heterogeneity, and allows us to focus in on what does this cluster do, what does that cluster do. Also, what it allows us to do, if we move to the very right-hand panel, is going from sort of the 0 to the 4, this is different levels of expression of, for example, CFTR. And what you can see is that, for example, at the ionocytes, the top may be expressing relatively high levels of CFTR. In the case of the ciliated cells, which we previously believed to be the predominant source of CFTR in the airway, we find very, very little expression. So it just shows the power of this approach. And if we go to the next slide, what I'm going to do is basically just sort of summarize the data that we have thus far. That for the ionocytes, that at least a good percentage of cells express high levels of CFTR messenger RNA. For the secretory cells and for the basal cells, we have a moderate level of expression. And finally, for the ciliated cells, we have very low or absent expression. So this is important because as we seek to develop genetic-based therapies, we want to sort of recapitulate, if at all possible, these levels of expression to better represent CFTR function in the airway. So how do we use this information? How do we use this understanding of cellular composition and heterogeneity to provide a foundation for development of effective genetic-based therapies? And for that purpose, we've put together an animation. Let's watch it together and learn something about how these processes of genetic-based therapies may be applied to the CF airway.
Promising therapies are being developed to treat the genetic cause of cystic fibrosis. When delivered through the airway directly to cells, these therapeutics have the potential to restore CFTR function in the lungs. Highly specialized cells that line the respiratory tract help protect airways from invading microbes, infection, and injury. At the innermost layer, basal cells are constantly replacing damaged and aged cells. Secretory cells release mucus into the airway to trap foreign particles, while ciliated cells move the mucus up and out of the lungs. Ionocytes are thought to express high levels of CFTR protein. The instructions to make CFTR protein are encoded in the DNA of each cell. The CFTR protein helps control the movement of salt and fluids into and out of the cell to keep mucus at a healthy level. With cystic fibrosis, DNA mutations create either abnormal or no CFTR protein in the cells, causing a buildup of thick, sticky mucus. By providing the correct instructions, it is possible to reprogram cells to make normal CFTR protein. Correcting CFTR expression in even a portion of the cells could be enough to restore sufficient function. Two potential treatments are RNA therapy and gene therapy. Both approaches can deliver a correct copy of the genetic instructions to cells in the lungs. With the correct messenger RNA or DNA sequence, cells would be able to produce normal CFTR protein that moves to the cell surface. There, it can adjust the amount of salt and fluids flowing through the cell membrane. Over time, the correct copy of the genetic instructions is degraded or lost as the cells naturally die and are replaced. Regular, repeated treatments will be needed to ensure enough CFTR protein is present to hydrate mucus. Gene editing is a potential therapy that would permanently repair the CFTR gene mutation in cellular DNA. The goal is to specifically target basal cells because they have the ability to divide and differentiate into other cell types to replenish lost cells. As cells die off, the basal cells with permanently repaired DNA generate new cells that contain the correct CFTR gene sequence. If enough basal cells are successfully edited, cells with normal CFTR will constantly be supplied to the airway surface. Repeat treatments would be rarely required, if ever. Cell-based therapy involves removing cells from the airway to create space, a niche, for the delivery of gene-edited cells. Cells are taken from the same patient, corrected via gene editing in the lab, and delivered into the airway, where they will generate new basal cells and specialized cells that create CFTR protein. Much like gene editing, this cell-based approach would permanently restore CFTR function for people with CF. Scientific progress in genetic-based therapies is advancing at an unprecedented pace. But the path to a cure is not a straight line. It will take time and investment to develop these treatments that could lead to restoring CFTR function in not just the lung, but in all parts of the body affected by CF, including the liver, pancreas, and GI tract. The Foundation is committed to ensuring that everyone with CF has a treatment for the underlying cause of their disease, and one day, a cure.
Well, I hope you enjoyed that animation. And what I'd like to do now in the second half of my presentation is really say, how do we think about applying these genetic-based therapies for restoration of CFTR function? As was indicated in the animation, really two methods at least are available. One is mRNA delivery, and the second that is of gene delivery or gene therapy. And what the schematic on the right-hand side of the slide shows is really the delivery through the mucus of cells directly into a cell of some kind of vector, which will contain either the therapeutic RNA or the therapeutic DNA. Now, Dr. Limberis is going to spend much more time in her half of this talk speaking about what types of vectors we can use for this purpose, but they can either be non-viral, for example, lipid nanoparticles, or they can actually exploit naturally occurring viruses. But in either case, the goal is to deliver that payload, whether it's the messenger RNA or the therapeutic DNA, into the portion of the cell required for restoration of CFTR function. In the case of messenger RNA, we really have to sort of unload the therapeutic mRNA right in the cytoplasm, where it'll be acted upon by the normal cellular machinery to give rise to the protein and then functional CFTR. In the case of DNA, we have to get it across the nuclear membrane into the nucleus, where it'll either become incorporated into the chromosomal DNA, and therefore quite stable, or maybe remain in what we call an extrachromosomal state that is still within the nucleus but not integrated. But in either case, we want that DNA to be converted into expression of the CFTR mRNA, and then protein, and finally channel function. So that's really the approach of delivery that's necessary to restore CFTR function. So how do we think then about restoring CFTR function to the CF airway? And just shown on the left here again is sort of a summary of the video where you see that thick mucus, which is basically compressed upon the cells. It's dehydrated, and there's no or negligible CFTR protein that's active. So what we would like to do is convert that situation to the one shown in the right-hand panel, where a certain percentage of cells, and we believe somewhere between 15 and 30% of cells that are actually expressing CFTR, will be sufficient to restore that normal chloride and bicarbonate channel function, to thin out the mucus where it's hydrated, and as well have the ciliary motion to sort of move pathogens and contaminants up and out of the airway. So how do we think about actually converting those CF cells to now, where shown in blue on the right is now they're beginning to express CFTR protein and function. And let's look at this for the three cases, the modalities we're talking about, that of mRNA delivery, gene delivery, and gene editing. So first, in the case of messenger RNA delivery, what you see in the lower panel on the left is that in the cytoplasm of the cells that are blue, you see single-stranded messenger RNA molecules that then can act to give rise to CFTR protein and to channel function. And from what we've learned from that single-cell RNA-seq analysis, where we really are focused on trying to restore that CFTR activity to, is the ionocytes as well as the secretory cells. the ciliated cells is sort of an open question, and we don't even know what is the consequences if we deliver CFTR protein to a cell like the ciliated cell that normally expresses very low or negligible levels. So that's something that we have to further pursue. But the idea is to really restore to enough frequency of cells that CFTR messenger RNA so it can give rise to protein. Let's now look at the issue of gene delivery. In this case, what you see is in the nuclei of the cells, we're showing sort of in a schematic, sort of delivery of the double helical DNA that will encode now the CFTR messenger RNA, and then the protein, and then the functional channel. And again, as I mentioned before, depending upon the vector that's used, either that DNA will become integrated in the chromosomal DNA or maybe remain sort of extra chromosomal. But in either case, the goal is that it would produce the protein and the function. Now, I would say that both in the case of messenger RNA and in the case of DNA, there's some question about how long the benefits, the therapeutic benefits would exist. In the case of messenger RNA, it's rather labile or short-lived. So it's unlikely that it's going to go on for months or years in terms of its benefit. We'll probably have to re-administer. And in the case of the DNA delivery, even in that case, we know that those surface exposed cells are regularly being lost as a result of inflammation or injury or infection, being replaced by the basal cells. So unless the basal cells themselves have been restored with CFTR function, it's unlikely that we're going to generate additional surface cells that are expressing CFTR. So anyway, there's tremendous opportunity, but also we just want to show how sort of the biology gained is giving us some idea of what we need to achieve. Finally, let me talk about this issue of gene editing. And really the importance of gene editing was highlighted earlier this month by the award of the Nobel Prize in Chemistry to two outstanding scientists who really discovered first what is the mechanism that bacteria use of this editing in an immune response, and that's now been exploited for introducing mutations or correcting mutations in a number of model systems. And that's in fact what's being exploited here for the purpose of essentially correcting the mutations in the CFTR gene. And our focus here is really in the basal cells. We want to develop a treatment that is lifelong. And in order to do that, we really need to correct the mutation in the basal cells so that they, being a self-renewing stem cell population, both as they proliferate, but also as they give rise to the differentiated specialized cells, will also now be expressing CFTR in its normal pattern of expression. Now, I would just say that the targeting the basal cells is not a trivial matter. They're protected by those surface cells. So there's a lot of work being developed how we can specifically target the basal cells. We may need to transiently remove those surface airway cells so we can get access to the basal cells. So no one's really demonstrated yet in sort of model systems the ability to effectively edit the basal cells directly in their normal anatomical position. But what has been done recently is to actually take airway basal cells from explanted lungs of CF individuals, to expand the basal cells from them, and then to perform two different types of editing. Shown in A is really site-specific correction. That is, this is a very specific correction of mutations such as G542X or correction of delta F508. And what's been shown is that those edited basal cells can now be put into conditions where they give rise to the ciliated, the secretory, and the ionocyte cells. And then when we assay, shown on the upper right, we can demonstrate restoration of CFTR function. Another approach that several groups are pursuing, including our own, is to see, can we develop methodologies that are more general, that can apply to the vast majority of CFTR mutations? And this involves essentially deliver a larger piece of DNA, but again, right in the natural chromosomal context with restoration. And again, we and other groups have recently shown that this can be successful. Finally, there's now approaches where people are using induced pluripotent stem cells, where we can start with skin cells and blood cells from a CF individual, can correct the mutation in these very, very powerful stem cells. And now there are protocols available to actually derive from them exactly these airway basal cells that we could use for regenerative purposes. So finally, I've spent the time in this talk focusing really on the airway. But of course, we know that CF disease affects many organs. And so the goal really is, once we have developed methodologies that work in the airway, the goal is then to begin to apply them, whether it's the pancreas, the GI tract, or other affected organs. So what I'd like to now do is turn the presentation over to Dr. Limberis, who's really going to focus on really what are some of the barriers to effective delivery of these novel therapeutics, but more importantly, what are some of the real advances that have been made in recent years that give us real hope that these types of approaches will be translatable to CF patients. Thank you.
Great, thank you, Brian. These are my disclosures as it pertains to the next half of this presentation. So I'm going to be covering the effort of the Cystic Fibrosis Foundation to actually develop next-gen gene therapeutics to treat all people with cystic fibrosis. As we heard, modulators may benefit a significant percentage of people with cystic fibrosis. Our goal is to develop an effective therapeutic for all people with CF, especially for those that do not respond to modulators. Cystic fibrosis is an ideal candidate for genetic-based therapies. On the left-hand side is a simplified chronological diagram from the discovery of the cystic fibrosis gene on the top left in yellow in 1989, and how quick molecular therapies were actually developed and tested in clinical trials. Within 3 years of the discovery of the gene, the first gene therapy trial was initiated in 1992. However, to date, there's no approved gene therapeutic for cystic fibrosis. Since then, though, there have been a lot of efforts that have been placed on the development of small molecules that will actually alleviate the phenotypical disease of cystic fibrosis. In 2012, with ivacaftor being approved, shortly after the combination treatment of ivacaftor, lumacaftor, and most recently in 2018, the triple treatment that was approved. Why is CF the ideal candidate for gene therapy? It's a single gene disease, and we know the gene well. And the lungs are large organs that are accessible to therapies. There are key issues for translating these gene therapeutics. And the main ones are that the cystic fibrosis gene is large, which limits the use of certain types of gene therapeutics. Central to the translation of these gene therapeutics is actually targeting the right cell type. As mentioned earlier, there are several different cell types in the airway. There are specific that need to be corrected to actually see a phenotypical change. The physical delivery of these gene therapeutics to the right cell types in the lung is not easily done. And when you're dealing with viral-based therapeutics, the re-administration in the setting of immune responses is difficult. So the three strategies that have been taken are basically to repair, replace, repair, or restore the cystic fibrosis gene. On the left-hand side is a double helix structure of the gene. In red is highlighted a CFTR mutation. The three different strategies are either gene transfer, where you're basically just replacing a normal copy of the cystic fibrosis gene editing, which is simply removing and replacing the portion of that gene that's defective, and RNA therapies. The potential of gene therapy is incredible. One example from bench to bedside is with a disease, a genetic disease known as spinal muscular atrophy type 1 on the left-hand side. Babies are diagnosed shortly after birth and within their first 6 months of age. And 75% of them will succumb to this particular disease by their first birthday. Babies will not achieve motor milestones. They will never sit unassisted. They will never crawl or walk. However, a gene therapeutic based on adeno-associated virus vector that we'll be discussing later in the talk, one dose of this particular therapeutic delivered to babies at the age of 13 months or younger allows for these children now to actually live longer and healthier lives and actually meet these particular milestones. On the right-hand side is a young lady that's running, meeting those motor milestones, even though she was diagnosed with SMA at birth. She can talk, achieve head control, they can roll over and walk, and importantly, live longer, healthier lives. The two categories of leading molecular therapeutics for CF are virus vector DNA on the top, and on the bottom, the lipid nanoparticle mRNA. There are advantages and disadvantages to both. For the viral vector DNA, the main advantage is that they achieve high and sustained expression. Some of these gene therapeutics can actually package the large cystic fibrosis gene as well as the promoter that's required to drive the expression of the cystic fibrosis gene in the airway cells. However, these particular gene therapeutics can be immunogenic, which makes redosing, which in fact for cystic fibrosis is necessary, quite difficult. Lipid nanoparticle encoding mRNA have the advantage of a rapid expression that can be effectively redosed. They can actually accommodate the large packaging size of the CF gene and the promoter that's required to drive expression, have a favorable immunological profile, but lead to short-term expression. However, this is overcome by their favorable immunological profile, allowing for redosing. So the common theme for molecular therapeutics and their development for CF as we progress into the translation in clinic can be categorized in three different groups. Optimization of the CFTR on the left, in the center, airway-specific formulations to actually be able to deliver something in the airways in a non-inflammatory, invasive manner, as well as the delivery devices. There are physical barriers to effective airway-directed gene therapeutics, and these are noted in the picture on the right. On the very top of the airway sits the mucus layer, and the mucus layer actually prevents the entry of foreign inhaled particles from the top of the airway surface into the cell. On the very top of the airway epithelium lie the cilia, which coordinate the mucociliary clearance, a mechanism by which the lungs clear anything that's inhaled and that's foreign and potentially damaging to the airways. The glycocalyx that sits at the very top surface of the airway epithelium is an electrostatic barrier. So its particular charge is such that special type of vectors may be expelled because of their difference in charge. Tight junctions, the airway epithelium is polarized, which means it has two distinct layers, the apical layer and the basolateral surface layer. And it's very difficult to gain entry to the basolateral surface where the majority of the receptors for these gene therapeutics lie when delivering a gene therapeutic from the air, from the airway. There are also biological barriers to effective airway-directed gene therapy. So even if we are able to bypass the physical barriers of the airway, once a gene therapeutic enters the airway cell, it can actually lead to immune responses on the left that can either be cellular or humoral. These result in the activation of antibodies against the gene therapeutic, the cystic fibrosis gene, as well as T cells to the cystic fibrosis gene and the gene therapeutic. So on the right-hand side, typically what happens is a gene therapeutic will transduce an antigen presenting cell, and based on the type of an immune response will either give rise to CD4 T cells or CD8 T cells. And through different types of mechanisms, they will give rise to either antibodies that will prevent readministration of the gene therapeutic, or T cells that will actually kill or destroy the transduced cell. This next slide, I'd like to highlight the effort of the Foundation in trying to identify molecular therapies that are promising and that have the potential to treat the underlying defect in those subjects that are not amenable to the treatment by the potentiators and correctors. This particular endeavor, initiated in November of 2019, was a very concentrated effort by the Foundation to actually meet with companies in the US and worldwide to actually identify those that have potential promising technologies that could be applied to gene therapy or gene therapeutics for CF. The Foundation committed more than $30 million to these programs, and it's actively looking at new programs and including these in this particular pipeline in an effort to identify gene therapeutic for all. So the next slide, I'd like to talk about this particular pipeline. There are four main categories for which the Foundation has focused a lot of effort and supporting companies. On the far left, we're looking at programs, 14 different programs, some of which are funded and some of which are under evaluation, that deal with restoring CFTR function in patients with either nonsense mutations and using particular technologies like mRNA oligos. The second column, working on gene delivery mechanisms, or gene delivery vehicles. There are 12 different programs looking at either viral or non-viral approaches, some of which have been funded and some of which are under evaluation. In the third column, gene editing is also an approach that the Foundation has been looking at and has actually got a program that is funded and 6 that are under evaluation. A lot of effort is also placed on developing models and tools that are required to actually allow for these programs to be translated, to allow for the programs to use assays to understand immunogenicity, functionality of the particular product that has been expressed, as well as other programs in the pipeline. The viral programs that are being supported by the Foundation, there are 3 currently, and 2 for adeno-associated viral therapeutics from a company known as 4D Molecular and Spirivent. And Spirivent actually has another gene therapeutic based on a virus known as lentivirus. So on the far right are just 2 different schematics of an AAV and a lentiviral vector program. The viral vector programs from 4D have to do with the molecular evolution of AAVs. So trying to take an AAV that has an established phenotype and evolve it in such a way that you create an AAV that has an improved lung tropism. And the initial program that was funded by the CFF, 4D actually did identify an AAV vector with improved lung tropism. And the Foundation has now committed to providing up to $14 million to actually accelerate this CF program, with the end goal being a phase 1 clinical trial. Spirivent is actually focusing on the development of 2 different viral strategies, as I mentioned earlier, based on AAV and lentiviral programs. They were recently purchased by Spirivent and then became part of a larger company known as Sumitomo. They focus on AAV for effective targeting of the airway, as well as lentiviral vector that results in lifelong correction of the defect. I'd like to highlight in the next couple of slides certain abstracts that have been accepted in this year's NACFC meeting. And this is the development of an airway efficient AAV that evades humoral immunity. In this particular strategy, what was done was actually identify on the left-hand side an AAV that has an enhanced transduction for airway that can actually evade antibodies that are circulating and can effectively transfer the genetic cargo into the lungs. Early data has shown that it has at least a fourfold improvement in resistance to AAV antibodies. And most importantly, on the far right, when you look at cross-sections of airway cells on the top trachea and on the bottom picture, the bronchi, where you actually see the red color denotes a positively transduced cell, suggesting that these are highly effective gene therapeutics. The next slide is an abstract from Arcturus, where they are developing a non-viral approach using an mRNA therapeutic that specifically targets the airway using a formulation known as LUNAR. And this preliminary work done in animal models, which is quite impressive, when you look at the top right-hand side, in panels A, B, C, and D, are airways that have been positively transduced with this particular formulation expressing CFTR. You're looking at the green signal, which is quite evident, or in panels E, F, G, and H on the right, you're actually looking for the dark colored transduced cells, showing an impressive transduction profile using this particular formulation. And in the graph is its translation in a relevant clinical model of CF, the CF mouse, and across the nose, these were actually formulations that were delivered. And when compared to a saline formulation that does not contain a gene therapeutic, noted on the left in blue, there's a clear difference between the blue and the purple bar, suggesting a positive transduction of that particular respiratory epithelium layer, further demonstrating the potential of this approach for clinical translation. The future is quite bright. What it holds is that the Foundation has set the path for evaluating several therapeutics in parallel in an effort to develop in a very time-effective manner the most efficient and safest molecular genetic therapeutic that can be effectively redosed. In parallel, there are efforts to develop model systems to better predict the translation of the efficacy of these molecular genetic-based therapeutics in humans, as well as assess in preclinical models the immune responses to both the actual vector therapeutic and the cystic fibrosis gene. Thank you. On behalf of Brian and myself, I'd like to thank you for your attention.